Should I stand anywhere?
Great.
It's okay? I just want to be cognizant of my time. Like, I know I can kind of drone on. Not really. I think people enjoy most of my stories. Maybe they're just being nice. All right, to reintroduce myself, my name is David Chichuka. I am a photographer since moving to New York City, but I wasn't always. I used to be a lab bench scientist, and this kind of presentation is sort of a year and a half worth of life's journey crammed into 8 minutes, so please bear with me. But to start us off, I was just going to give you guys a sense of my past life, which was as a microbiology researcher. My life consisted of microscopy images and these tiny little cells. I was doing work with biofilms for the uninitiated. Those are small microbial communities that end up forming these masses like this. This is a yeast, a species of yeast. And, you know, lab work day after day, 12 hours a day sometimes. It's grueling. It was great. I love science. I've spent my whole life doing science. But something else was kind of like bubbling up underneath. These are just like more samples. Even in my like documentation of my own sort of lab journals and stuff like that, I did my best to kind of make them at least kind of nice to the eye. There was a sort of underlying creativity that I hadn't really tapped into until I started to think about other things to do outside of the lab. I kind of became jaded with the pursuit of these, what can sometimes be esoteric questions, right, when it comes to research purposes. Not always. I personally look for the value in anything that we're doing. But I just found it hard to convey that importance to a lot of other people, and I knew that other scientists had that same sort of problem. A lot of people don't know where money is going when it goes to fund research, and so I started to explore a new message, which was like looking at the scientists around me, the students around me, asking them what they did, why they did it, and to share that. And I started photographing them, coming up with a new idea to document scientists, share their stories. That's kind of an ongoing project that's kind of been shelved because I have to wear a lot of different hats. So I left the laboratory about a year and a half ago, and I started to try and improve my photography skills. You know, meeting up with random creatives in New York City, taking their portraits, speaking an entirely new language, because the language of scientists is, you know, way, way far away from any sort of creative talk as we kind of traditionally know it. So I met with people. Another new skill, as scientists, you're kind of doing your own work by yourself oftentimes. You have lab mates and stuff, but it all is on you versus talking to strangers, making conversations, helping people feel comfortable in telling like what they want to share. Those are all things that I've been able to kind of pick up since moving here. And like being introduced to this new changing environment. So it's not just the people, it's not just the lab spaces. Like when I came back from school, I started to explore other informal learning spaces. Like this is the Science Barge in Yonkers. I'm from Westchester. I live in Queens now. But other places like this, there are science communication events all around the city. Astronomy on Tap, Taste of Science, Biology on Tap is a new one. All these little spaces where scientists are actually trying to communicate with the broader public. So I started to explore these spaces to see like what people were doing, whether it was presentation style, what kind of events they were throwing, what kind of people were coming, and how do all these things sort of interact, and like where do I make my own little space here. So eventually, shout out to Genspace, I found a little home, a little studio, a living studio in more ways than one. And I'm eternally grateful for the opportunity to be able to come here and explore this new passion of mine and just figure it out. And what Genspace has sort of offered is my first role here was as the science communication ambassador, so documenting the classes that were going on, the people that are coming here, the stories that we could tell outside of this laboratory space. And looking at science, and not so much in just like, look how crazy and cool this photo is, it's also like, who are the people that are doing it? Who are the people that are showing up? Like, why are they here? Why should you come here? All those sorts of things. And fast forwarding to the latest thing that I was fortunate enough to work on, after the science communication ambassador progress was completed, I was invited to help with an after-school program called BioRocket. In this program, we basically teach high school students some molecular biotechniques, things that we constantly remind them are things that most people don't know until your second or first year of grad school. So very, cutting edge, very new, very novel to a lot of these students who, although these are very high achieving students, the fact is you're not going to get exposed to this kind of environment until way down the line when people feel like you're ready.
What would be an example of that?
What do you mean? Like what kind of activities we did?
Mm-hmm.
So as far as like genetics and stuff go, it is like lower tier or basic stuff like PCRs, sequencing, genes, transforming bacteria, changing genes around, switching genes around, being able to kind of identify those sorts of things and the processes to learn that. And so my background helped because I kind of have an understanding of what all those things are. Beth really ran the show. She's the brains of the operation. And I kind of found myself more as like a documentarian. These are more of the things that I was able to capture. And more as like, almost like a participant, because in this informal learning space, it's not so much like we're here to teach you something and you have to know it exactly how we teach it to you. We kind of have a different way of doing things in that like you are here to be engaged. We are here to help create an environment where everybody can collaborate with one another. You can kind of learn something about somebody else. We were more focused on building a team, building a community, because Genspace at its core is ultimately a community biology laboratory. It's not just about the things you can learn here, but the people that you can meet and the ideas that everybody can exchange. So kind of bringing it back to the photography, is like, how do you capture that? It's not just a slide. It's not just a photo of a slide. It's not just a photo of people doing lab work, although that is part of it, and it's nice and it's flashy, and we get to show everybody what's going on. I'm still kind of processing and digesting all the things that we were able to do this summer, this spring, but ultimately I kind of want to follow this along and peel back those layers. Like, yes, we're teaching you science. Yes, you have an opportunity to learn different things here. But more important than that, like, what connections are you making, both to the things that you're learning and to the people that surround you? And what does that look like? Ultimately, what my work sort of boils down to today is like, what does a community biolab look like? What kind of people are here? What kind of science or knowledge can it offer? What kind of questions are coming up? And if I had to kind of pick an overarching theme between the science and this art, as far as like the reflections I've had and things that I've found, is as you become a better scientist, you learn to ask better questions. And as I've sort of done this creative thing, and I have a hard time calling myself an artist or anything, but as I dive deeper into this creative and this artistic world, you also start to ask better questions. It's not just who's here, it's why are they here, and what are they taking away. And that's kind of just a little bit about what I'm trying to do. So if you want to see more of my work, I'm largely on Instagram and some other places. So that's my handle. And the last question I kind of pick here is, how do you express your authenticity? Because another big theme between science and art and photography specifically, I went to a talk recently and somebody was talking about how photography is always, or as it evolved, has always kind of kept this element of truth to it. Like when it was first developed, it was hard to doctor a photo, right? It was hard, there wasn't any Photoshop back then. So if you got a photo of something when photography was first sort of emerging, it was sort of the truth. And a lot of people consider science the truth, in that we seek the answers to specific questions. But today, those truths are a lot muddier. You can Photoshop images. You can cherry pick studies. You can cherry pick data. So like, what is truth? And the only thing that I can really come up with is authenticity. Like there could be an absolute truth, but in reality, there's a lot of little truths. Like we all speak our own language, we all speak our own stories, our own little truths. Each of these studies have nuggets of information and wisdom that can teach us something. And so I ask everybody here, and sort of my work in general, the things I sort of try to capture is like, how do we express our authenticity? Because if we can be authentic, then you're speaking your truth, and somebody can trust you based on that truth. And outside of that, like, yeah, I have no answers. So that's me.
Thank
Yeah.
you.
No, cool. Thank you. All right. All right, so I'll try to keep this sort of tight, but I'm going to start with a bit of a story. First, I'll introduce, I'm one half of Epicurean endocrinology, so I'm the Byron Rich hat. I'm assistant professor of art and affiliated faculty in computer science and director of the Art Science Innovation Program at Allegheny College, which is a little fancy liberal arts college north of Pittsburgh. So I want to start with a little story of when I first moved to the United States almost 8 years ago. When I first moved here, I met a wonderful person who I would later marry. And when we first met, she had to go get a new prescription for birth control. And I was like naive and Canadian. And I was like, okay, that's cool. This is easy, right? We'll go to the pharmacy, we'll go to the doctor. This will be like, 45 cents at the pharmacy. It'll be super easy. I did not realize anything. I was so naive. So I didn't realize just how like politically difficult the institutions of control that kind of like presented a challenge to her just getting it from her workplace insurance, like the politics of the United States. And that kind of like blew my mind. So in my naivety going to graduate school, I'm like, I wonder if I could just make birth control, like an open source birth control bill. So that's what I set out to do. And I did this like with zero awareness of like my like white Canadian straight male like savior complex was, right? That I was gonna just set out and like solve this crazy problem. So I, of course, I was naked. I totally set out to do this. And what I did is I started by using cholesterol from a chicken's egg and synthesizing like the components to actually produce like hormone birth control. And I got pretty far. Getting the cholesterol was easy, but actually like doing the next step, like synthesizing like these key components, it was really hard. It took a long time. It relatively worked out, which was cool. But what the big problem was is that I was just like not anywhere ready for like the cultural like importance and possibility behind doing work like this. I was not a feminist theorist. I had no idea. I was just doing this because I could. Like I thought that I could like solve this problem and I was going to set up to do it. So I needed a crash course in feminist studies. And I was pursuing a couple other projects, so that had to like go on hold for a little bit until I had the time to do this. Fast forward a couple of years, and I met my wonderful former collaborator, Mary Magic, who I worked really closely with for a number of years on a project called Open Source Estrogen, which ended up becoming winning a few prizes and was like quite a relatively well-known project. I feel super proud to have worked on it. Out of that project, I started to work with Liz Flintz because I was fascinated by the overlap that I was seeing between the kitchen and the laboratory as these like highly gendered spaces, like just unbelievably gendered spaces. And the like historical baggage that came with both. But both were like so full of experimentation and so full of play and wonder and discovery, yet they were just immensely gendered, and there were like ideas around who was supposed to be in what and who was supposed to not be in what place. So we started to work on this project where we pursued the development of a protocol where like citizen scientists could actually like extract endocrine disruptors from their food systems and water systems. Endocrine disruptors being compounds that can bind with estrogen or endocrine receptors and kind of like mimic various compounds. So what we're focused on right now is estrogen. The reason we're focused on so much on estrogen is it's been going on for a long time, but there was a huge moment in about 2011 when Newsweek ran an article about like, and I'm going to use this phrase with full knowledge of like its difficulty and baggage around it, but intersex frogs, and intersex frogs being the result of hormonal birth control, women taking them, urinating, and that passing into like water systems. This is not the case whatsoever. Like that is such a minute portion of what actually like causes endocrine disrupting compounds to like proliferate our food system. It makes up like 0.01% of like the entirety. What it really is like atrazine, for instance, which is a really commercially available and super common herbicide that's used on corn, sorghum, all sorts of places like that. In fracking, a lot of the things that they inject into the wells, they actually like extract natural gas, immense sources of endocrine disrupting compounds, especially estrogenic compounds. So this like sex panic that frogs were turning female because of women taking birth control and passing it through was just more like kind of an apparatus of control, you know. And we found this like obviously profoundly troubling, as I'm sure everybody in this room. So what we wanted to do is develop this system that people could actually like extract endocrine disruptors from their food and water system. So we came up with an Epicurean endocrinology. So what we're working on right now is developing this like almost a Blue Apron style food box system where we send you all the tools to actually extract endocrine disruptors from food and water sources. We also send you recipe cards and some foods to test and also make molecular gastronomy recipes using these foods. So if you want to think about it as like this very speculative and totally not like working like kind of like act of biotechnical resistance, in that like maybe you could make these like highly endocrine disruptor rich foods and you could actually like alter your own like body by doing this. Of course, that's not practical. The reason why we do it, I'll get to the reason at the end. So this is what the cooking sex, Epicurean endocrinology cooking sex lab actually looks like. So it's a hybrid kitchen lab space where we have set up all the tools and technologies to do the solid phase extraction protocol to pull these endocrine disruptors out of your food. And then we use something called an ELISA assay back in our lab to actually find out like the concentration in that has like come from people's foods. So you'll be able to send your sample back to us. We'll test it and give you data on like exactly what was in it. So this is what it looks like set up with the ads and everything in it and all the tools and technologies. The micropipette, by the way, is just for show because it looks cool. We don't actually really use it. We use it sometimes in the essay, but you know, we gotta like play the part of this is just like so just so you know. 3 minutes. Okay, so this is set up at my like home gallery, just so that I could like hopefully, or Liz and I could hopefully get shows with it. So this was like the first iteration. There's now 4 of these labs in various places. There's a mini one back here actually in Genspace. There'll be another one in Flex Factory later this month, and if you want to do a workshop with us, that show will be opening on the 23rd of June. We'll actually be doing endocrine disruptor workshop extraction lab here at Genspace. Yeah, so thanks for having me. So this is us testing pickled dilly corn. I live like way out in the country in the middle of like real America, as lots of people like to put it. So we test a lot of things that we find like in that region because there's like in this kind of like middle America, and I live almost on the like New England side of things debatably, but whatever. We're fascinated by this like idea of middle America and middle America being obsessed with like profitability and like high production of like corn and whatever. So they're using these endocrine disrupting compounds like atrazine to promote crop growth, which stands like diametrically opposed to their value system, like on the far right in terms of like, and I'm gonna use this very carefully again, like intersex as Newsweek would put it, people, or people that don't conform to their like gender norms. So we find that like profoundly fascinating, right? That they're making like this proliferate like our food and water systems. So these are a couple of projects that we've done, some of the recipes that we're doing. These aren't molecular gastronomy recipes, but these are like feminizing and masculinizing meals that Liz put on in Craig Garden in the Adirondacks, which were really great. And we'll use some of these recipes actually in our food box system once we get it up and running. So again, this is like our play mad scientist with your food. which is kind of fun. So this is the little syringe and the solid phase extraction cartridge, called C18 cartridge that you use. We also developed, Mary and I a few years ago, a column chromatography method where you can actually roll your own cigarette filters and actually extract endocrine disruptors using that, which is a really interesting process. This is what our little recipe cards and the solid phase extraction protocol card look like. You can grab one back there if you're at all interested. This is the current setup at MOCA Tucson. We just did a series of workshops and set up a thing put on by a wonderful curator named Alex Young. The show is called Broken in the Dark, which Mary's also in, and I highly recommend if you get a chance to look into it. Oh, that's interesting. That's not me. So the point of my work, this is the last one. That's my thought. The point of the work, a lot of times people our big worry is that people get their hopes up that we can create these make your DIY, these DIY hormone therapy or birth control, and that they're going to be able to use these as an emancipatory technology. That's not the point though necessarily. The point is to say, why the fuck should some goofy artists have to come up with these biotechnical pieces of resistance? Why isn't this just available? And so that's the larger point of both these projects, and that's kind of what we're aiming for. So with that, I'll end. Thank you very much. Oh, don't take these. They're super expensive. You need them for the workshop, but you can take these.
Yeah.